Hello, this is Mr. Wiersma. This is my second presentation on PowerPoint with video, and this one's on meiosis. Now remember last chapter we did mitosis, which resulted in two identical cells which were diploid, which in a human we have 46 chromosomes. The goal of meiosis is to form haploid cells, and if you remember that term from the previous chapter, those are cells with half the number of chromosomes as original. The goal of haploid cells is to make gametes, which will be used in sexual reproduction. So where does meiosis occur in us? Well, it's pretty obvious. They occur in a male's testicles, resulting in sperm, which are haploid, and they also occur in a girl's ovaries, which result in eggs, which are haploid. And remember, the haploid amount in humans is 23 chromosomes, so when you have a sperm and an egg combined, then you get the full number of 46 again. The first stage of meiosis is prophase 1. In meiosis, they designate whether it's the first time through the cycle, the second time through, so they do have Roman numerals after the name. So this is prophase 1. And if you remember the cell cycle, it has 3 steps: interphase, the nuclear division, and cytokinesis. Well, before meiosis begins, just like mitosis, the DNA doubles, and so when we start meiosis, specifically prophase 1, the chromosomes now have been duplicated, so they are in pairs. So we have 23 pair in a human, 23 pair of homologous chromosomes. If you look at the little picture here to my left, you'll notice that in this picture there are 2 pairs of homologous chromosomes, and they get side by side during prophase 1. This is a key part of meiosis because when they are side by side, it's called a tetrad because there are 4 chromatids, and tetra means 4. But when they get side by side, they exchange little pieces of their chromatids with the other homologous pair. And so this creates genetic diversity. And if you look at the picture, a little chunk of the blue switches with the purple, a little chunk of the orange switches with the red. And so again, this creates a tremendous amount of genetic diversity. It's called crossing over. So when I ask you on the test when crossing over occurs, say prophase 1. Okay, this is showing you metaphase 1 of meiosis, and it's similar to mitosis in that the chromosomes are at the equator of the cell. They line up towards the middle. But one thing you might notice is that here the homologous pairs are side by side as opposed to directly in a row underneath each other, and that is crucial too. But if you look closely again, you can see the crossing over from the previous step because there's little color differences in each set of homologous chromosomes. So this is metaphase 1 of meiosis. In anaphase 1 of meiosis, the chromosomes are pulled to opposite sides by the spindle, which recall from last chapter is made by the centrioles. And a spindle attached to the centromeres and pull the homologous chromosomes to separate sides. But notice that the chromatids do not pull apart. The centromere does not break. The whole double-stranded chromosome is pulled to the right and to the left. That is another difference between anaphase 1 of meiosis and anaphase of mitosis. Now, something else critical can happen during anaphase 1. It's called shuffling of the chromosomes. And I guess what that means is that when the chromosomes are pulled to opposite sides, they can switch around. And so in the picture, the orange could have gone to the right and the red to the left, or the purple to the left and the darker blue to the right. So they don't have to always, follow this exact pattern. They can shuffle around. And again, this creates a tremendous amount of genetic diversity too. In telophase 1 in cytokinesis, the chromosomes now have reached opposite poles. The nuclear membrane reforms, and so we have 2 cells that are different genetically, but notice they have 2 chromosomes that are both double-stranded. And again, notice the colors in each because of crossing over that occurred during prophase 1. The cell membrane pinches, the cytoplasm splits, and as a result of telophase 1 and cytokinesis, we have 2 cells. Starting with prophase 2, we're going to have 2 cells now, because in telophase 1, remember, the cytoplasm divided and the cell membrane pinched, forming 2 cells. But a key difference in prophase 2 that distinguishes it from prophase 1 is the DNA does not double this time, so the genetic material is the same as we ended with in telophase 1. And so if you look at the diagram, we have 2 cells. The spindle is beginning to form from the centrioles, and this will begin the second meiotic division. This is anaphase 2. If you look at anaphase 1 in the top diagram, notice that the chromosomes went to opposite sides of the cell, but the centromeres did not break, so they're both double-stranded chromosomes. But in anaphase 2 now, the centromeres do break, and so the chromatids separate at the centromere, and each chromatid goes to the opposite side of the cell. And so now, if you can visualize, we have 4 single-stranded chromosomes in each cell. Finally, we have telophase 2 and cytokinesis, the last step of meiosis. As you can see, comparing telophase 2 to telophase 1, now the cell membrane pinched again, and so we result in a cytoplasm divided 2, and so the result is 4 distinct separate cells. Notice that each cell has different colors of chromosomes in them. That indicates that they are genetically different, and they are haploid. They have 1 of each chromosome. And so the goal, remember, of meiosis is to create haploid sex cells, and that's what we get as a result of telophase 2, 4 distinct haploid, genetically different cells. And so that is meiosis.